Today, we begin our dives at a seamount in northern Costa Rica on the Pacific side. Our goal is to assist researchers with tissue sampling on adult bull sharks. Each member of the team has a job. Photographers and videographers document the sharks while others measure and take tissue samples. Everyone is on the lookout for a shark to observe, and the dive master watches out for the team. Our first dive begins at a cleaning station where bull sharks have been found in the past. Our expectation is to find the sharks moving slowly, allowing the smaller fish to remove parasites from their skin. The tool used for tissue sampling is attached to a pole with an elastic band. The band helps power the tool into the tough skin of a passing shark. Many of these sharks are between 2 and 3 meters or 6 to 9 feet in length. As the opportunity arrives, the diver paces the shark and launches the rod to gather the sample. What remains inside the tool after contact is a small sampling of the shark's skin tissue. The samples are collected, sealed in a lab tube, and eventually sent to a lab for analysis.
Isotope analysis with the tissue sample. That lets us know if the animal has been feeding in the area or if he's coming from a different location. And with enough studies, we can also know which location it's coming from. So that tells us like residency, if that animal's been there for a very long time or if he just moved in. Then of course we also do the DNA analysis. This is used first of all to see how related these sharks are to other sharks in the area. And more importantly, it's to study site fidelity, because we know that sharks, or certain species of sharks like hammerheads, are philopatric. It means they come back to their natal site, like sea turtles. Sea turtles go back to their natal beach. We know hammerheads and tiger sharks go back to their natal wetlands. So we're trying to figure that out with bull sharks too. It's called natal homing. When it does occur, then the population becomes distinct from others. And then you can say this shark comes from Coyote River, this shark comes from this other wetland. And then we could take a tissue sample from a shark, let's say in the Bad Islands, and we could be able to say, this shark really comes from Coyote. That's what we can do with the natal homing.
The reasoning for sampling and documenting these species is simply for their protection and conservation.
Establishment of management, creation of marine protected areas, expansion, and also to develop sustainable fisheries management tools.
As citizen scientists, we are able to take part in the protection, conservation, and regeneration of fisheries. Our assistance not only promotes local healthy ecosystems, ecotourism, and sustainability, it also promotes healthy ecosystems and sustainability globally.